I will start in a couple of minutes. I'll start in 2 minutes. So I'll share the slides. Okay, so this is the 8th live session of this course Structural Biology. In this, I'll be going through the lectures of week 8, which will basically be covering different aspects of microscopy, starting from the very basic light microscope going up to cryo-electron microscopy basics. And then, as always, I'll be solving some practice problems on the topic. So the first thing is that there are some limitations of light microscope. The naked eye, as you know, cannot see objects less than 100 micrometer in diameter. So you cannot see cells by naked eye. You can only see maximum up to the different micella of fungi. Beyond 100 micrometer, you need the help of microscope. There can be simple hand lens which provides magnification of 2 to 10 times. They are technically the simplest microscope. And then you have the common light microscope, which basically uses visible light and a system of lenses to magnify the images of small samples. So imagine you have an object here. You have one ray that passes through the lens undeviated and reaches the eye, and another which passes through the lens and gets refracted. And when you draw these lens back, then they meet at a point where you get the virtual image of the object. This is called the virtual image because it is formed by when you draw back the lens. So when the light rays that are meeting the eye, when you draw them back virtually, you get the image. That's why it's a virtual image. If the image had been formed by direct intersection of the light beams, then it would have been a real image. So this is how, from an object this tiny, you get a virtual image this large. This is the basic concept of the light microscope, which uses visible light, and it can magnify objects up to around 0.25 micrometer. So you can see bacteria, eukaryotic cells, up to mycoplasma using the light microscope. This simple microscope uses a lens or a set of lenses to enlarge an object through angular magnification alone, giving the viewer an erect, enlarged virtual image. It's called erect because it is in the same orientation as the object. It is not flipped or anything. It's enlarged very clearly and it's a virtual image I've already discussed. So this kind of simple microscope, that is the light microscope, can provide magnification, you can see, up to around 0.25 micrometer. And the magnifying glass, which uses a single convex lens, that provides magnification only up to 2 to 10 times. So these are some very basic instruments that have been used to enlarge and view very tiny objects. Now, a more complex version of the light microscope is the compound microscope, which uses an objective lens close to the object being viewed to collect light, which focuses a real image of the object inside the microscope. So let us look at this basic instrumentation of the compound microscope. So you have an object here, two beams of light. They are coming from some light source behind the object. They are passing through this objective lens and they are getting focused here. So here you have a direct image formed. It's a direct image because it is formed directly by intersection of these light beams, and the image you can see is flipped. So here the object was pointing this direction, the image is pointing this direction. So this object is forming a real image or a direct image of this object is being formed inside the microscope. If you imagine the microscope, so these lenses are all part of the microscope. If you imagine a tubing here, so if you imagine the microscope tube going like this, then the image is being, first image is being formed inside the microscope. This image is then magnified by an eyepiece lens that gives the viewer an enlarged virtual image of the object. So you have this image. Now this image is being magnified by the second lens, which is called an eyepiece lens. Here you can see in the eyepiece lens what happens. The light passes through the image and goes to the eye, but there is no direct intersection of the light beam. So the image formed is not a direct image, but a virtual image. And how you can form that image is that the rays that are meeting the eye, you draw them back virtually as dotted lines, and wherever the rays are meeting, that is where the virtual image is being formed. So the second image you can see is much more highly magnified compared to the first image. So the compound microscope, which uses both an objective lens and an eyepiece lens, causes a much greater magnification compared to the simple microscope. And you can see here first a real image is formed, which is the image 1, which is only slightly magnified compared to the object. But the second image that is formed, which is image 2 here, is highly magnified. So overall, the image that the eye is observing is this highly magnified image. And that is why the compound microscope is much more better than the simple microscope for viewing tiny objects. Now, there are some aberrations that can occur in a microscope. One is a spherical aberration. Here, what happens is that when the light rays hit the periphery of the lens, they are refracted more compared to the light rays hitting the center of the lens. You can see the light rays hitting the center of the lens are getting concentrated in a particular point. But the light rays from the periphery, they are not getting concentrated in the same point. Rather, they are meeting somewhere earlier. So what this will do is it will cause a blurry image. If all the light rays are focused at a single point, then the image will be sharp. Otherwise, there will be a blurry image. So spherical aberration causes a blurry image, and it is because the light rays striking the periphery of the lens are getting refracted to a higher extent compared to the light rays passing through the center of the lens. Another kind of aberration is called the chromatic aberration, where you have white light passing through the lens and each wavelength in the white light is being diffracted to a different extent. So white light is passing through the lens, but after passing through the lens, it is splitting up into the different colors or components of the white light, because the extent of refraction for each wavelength, each color is different. So again, what this will do, this will cause a very blurry or unsharp images, and the edges of the image will be colored. So this is another deficiency of the microscope, where chromatic aberration is causing a loss in the resolution of the image. There are some other kinds of microscopy like confocal microscopy, which has attracted wide attention as an alternative to X-ray crystallography or NMR spectroscopy for determination of structure of macromolecules without the need for crystallization. In confocal microscopy, what happens is that you have a sample here and you have an objective lens. So light passing through the objective lens, it will reach the detector, but a part of the light before reaching the detector will be split by a beam. So imagine we start from here. You have a source of light here and it is passing through a pinhole. Now the pinhole, whatever light is passing through, it reaches the beam splitter. And the beam splitter splits the light up into two places, one which reaches the detector and one which goes to the objective lens and is focused on the sample. So like this, you can observe the sample and you will have a baseline where the light is not passing through the sample. So this is basically just another kind of light microscopy. We saw this light microscopy where there was an objective lens. The same thing is happening here, except there is a beam splitter to give you a baseline image where the light is not passing through the sample. Here it is said to be in focus because you can see the light is being focused at a single point on the sample. If it is out of focus, you can see here the light is not focused on a single point in the sample. It is focused at a point earlier, and on the sample there is kind of a, you can see this light is falling over a wider region of the sample. So you will get like a blurry image. Only when light is falling and being focused at a single point, you will get a sharp image. Otherwise, you will get a blurry image when it is out of focus. That is, light is falling in a wider region. So confocal microscopy is just another kind of light microscopy where you have a beam splitter. So you have the image, the light reaching the sample and forming an image, and you also have a baseline where light directly goes to the detector and does not pass through the sample. So this gives you a baseline. The most advanced kind of microscopy, which we will look at in this class in details, is the cryo-electron microscopy or cryo-EM. Electron microscopy, this technique, cryo-EM, is applied on samples cooled to cryogenic temperatures and embedded in an environment of vitreous water. So what vitreous water means is that the water is completely frozen, not as crystals, but as it is in the protein environment. So imagine you have a solution of protein in water, and the water is occupying certain cavities in the protein. It is helping stabilize the protein structure. Now, if you cool the water at normal temperatures, like not cryogenic temperatures, and you cool the water very slowly, then the water will end up forming crystalline ice, which will destroy the native structure of the protein. But if you suddenly cool the sample to cryogenic temperatures very fast, that is flash freezing, then the water will freeze in the orientation it occupies in the native protein. So the 3D structure of the protein will not be affected by the freezing of the water. That is why it's called cryo-EM, because you're using cryogenic temperatures. So how it is done is that an aqueous sample solution is applied to a grid mesh. So you have a grid like this, the sample is applied on the grid and plunge frozen in liquid ethane or a mixture of liquid ethane and propane. So you have a container having either liquid ethane or a mixture of liquid ethane and propane, and the sample is taken on the grid and plunge frozen means that very quickly it is just plunged into the mixture of this liquid ethane and immediately taken out. So the water that is there, it freezes in its orientation. It does not form any crystalline ice. It freezes in the native orientation of the protein. After that, data is collected and then different functions are done on the data, which we will discuss. So from each data point, you get a 2D projection of the molecule. And putting all the 2D projections together, a 3D map of the molecule can be reconstructed. These are basically done by different softwares. And ultimately, you get a 3D structure of the macromolecule. While development of this technique, the cryo-EM, began in the 1970s, recent advances in detector technology and software algorithms have allowed for the determination of biomolecular structures at near atomic resolution. So cryo-EM has advanced so far in recent times that now you can find out the structure of different proteins at near atomic resolution. So you can even resolve individual atoms. So now we will look at some terms related to the microscope. One term is resolving power. This is the power of an objective lens to separate distinctly two adjacent points. So suppose you have two adjacent points and the microscope can separate between them. So they are appearing as separate points. Then it will be called well resolved. If the points are starting to merge, it is called just resolved. And if it cannot separate the 2 points and you get 1 point, so instead of the 2 points, you will get an average point somewhere between the 2 points, then it is said to be not resolved. That is, the microscope cannot separate between these 2 points. So resolving power, the mathematical the expression of resolving power is lambda by 2Na, where lambda is the wavelength of the light and Na is the numerical aperture of the lens. So let us look into what is numerical aperture. Now, numerical aperture characterizes the range of angles over which the system can accept or emit light. So if you have a lens here and you have light passing through the edges of the lens and getting focused at a certain point F. So numerical aperture is defined as the ratio of the diameter of the lens, that is this D, divided by the focal length, that is the length from the middle of the lens to the focal point. So D, that is the diameter, divided by F, that is the focal length, is the value of numerical aperture. And it can be decreased by decreasing the amount of light passing through the lens. So suppose this is a large lens which allows a large amount of light to pass through it. Diameter of this lens is high, so numerical aperture will also be high. You can see it is 0.75. Comparatively, the lens on top, it is a small lens. It is diameter is lower and it is allowing less light to pass through it. You can see it is only allowing this much light to pass through it. Because the diameter is low, numerical aperture will be smaller. You can see it is only 0.25 compared to 0.75 of the lowered lens. So by decreasing the amount of light passing through the lens, you can decrease the numerical aperture. Another way of expressing numerical aperture is n sine theta, where n is the index of refraction of the medium in which the lens is working, and theta is the half angle of the maximum cone of light that can enter or exit the lens. So you can see the cone formed by the two red lines is the maximum cone of light that can enter the lens. And if you, this cone angle that is there, if you take half of the angle, that is theta. So you take sine of theta and multiply by n, which is the index of refraction of the medium in which the lens is kept. So that is another mathematical expression for numerical aperture NA. So NA can have two mathematical expressions. One is d by f, that is diameter of the lens divided by focal length. Another is n sine theta, that is the index of refraction multiplied by sine of the angle theta, that is half of this maximum angle formed by the cone from both the edges of the lens. There is another term called magnification, which is the process of enlarging something only in appearance and not in physical size. So that is what happens in a microscope. You can see here you have the outline of a compound microscope. So you have the object here. The rays of light passes through the objective lens and a direct image is formed inside the microscope. Now, this is image 1. Now, rays of light pass through the eyepiece lens and meet the eye. And if you draw these rays back, they will meet at a virtual point and you will get a virtual image, which is the image that is seen. This image is said to be magnified because you are enlarging the, so the object size was only this small. You are enlarging the size of the object much greater, but it is enlarging only in appearance, not in physical size. You are not changing the size of the object, but through the microscope, it is appearing to be much larger. So this is called magnification. The ordinary microscope is called a bright field microscope because it forms a dark image against bright background. So this is an ordinary microscope also which forms something called bright field image. You can see that the field is bright and the image that is formed is dark. So how this happens is that, as you know, light passing through a condenser lens. So that is condensing the light on the specimen. And then from the specimen, it passes through an objective lens. And from the objective lens, it passes through an eye lens or ocular lens and reaches the eye, which is the basic buildup of a compound microscope. Only this additional condenser lens is added. Condenser lens, basically what it does, so from a source of light, light is coming in all different directions. Condenser lens will basically focus all the light on the specimen so that you can see the specimen brightly. So this forms the bright field image, that is dark image against the bright background. This bright field microscope can be adapted as a dark field microscope by adding a special disc called a stop to the condenser. So in dark field microscope, how it will appear is that there is a dark field and the resulting specimen is bright. So here there was a bright field and the specimen was dark. Here there is a dark field and the specimen is bright. So how this works, the setup is exactly the same. Only something called a stop or opaque disc is added between the condenser lens and the light. So here what happens is that opaque disc is blocking all the light from entering the objective lens. Only the light at the periphery, you can see the disc will stop all these rays and only the rays at the periphery of the light will enter. And so this peripheral light is reflected off the sides of the specimen and you can see specimens where you can see this circle. Imagine this is a cell. So you can see only the sides of the cell brightly. The rest of the cell will be dark. This is because this opaque disc or stop is stopping the light from passing through the center of the specimen. Only the light at the periphery is passing through the periphery of the specimen. And so the periphery of the specimen appears bright. That is, the cell membrane or cell wall appears bright. Everything else is dark because the opaque disc is stopping the rest of the light. So this is called a dark field image. You can adapt a bright field microscope into a dark field microscope by just adding this opaque disc. Now we will look at why electron microscope was needed. Basically, electron microscope was invented because of the drawbacks of light microscope. In a light microscope, light passing through the slits gets diffracted. So here you have a light microscope. There is a condenser lens which is condensing the light on the specimen. Now, while this condensing is happening, there can be a diffraction. Diffraction means that the ray of light, instead of being a single ray passing through the lens, it breaks up or scatters into many different rays. So the light is getting diffracted, and the convex lens is collecting all the diffracted light. So all this diffracted light, they will get focused in different points, and it will form the image which is very blurry. So instead of getting a sharp image, the light is getting diffracted into many different rays, and each of these rays will condense at different regions of the detector. Wherever there is constructive interference, you will see a bright light. Somewhere there is destructive interference, you can see here, none of the rays are coming, so no image will appear. So what will happen is that a lot of blurry images will be formed. So I was talking about the drawback of the light microscope. Basically, in light microscope, diffraction can happen. Here you have a light microscope where the light is passing through a condenser lens, which is condensing the light on the specimen. Now, if this was an ideal case, like shown here, the light on the specimen will simply pass through the objective lens and then the ocular lens and come to the eye, where you will get a focused image. But in reality, what happens when the light passes through the specimen, it gets diffracted. That is, it will scatter in many different angles. And as the width of the specimen increases, the diffraction angle will also increase. And so when it is getting focused by the objective lens, instead of focusing at one particular point, it will focus in multiple points, forming a very blurry image. So that is the drawback of the light microscope, is that instead of a sharp image, you are getting several blurry images. To combat that, the electron microscope was invented. Basically, the principle of the electron microscope is the de Broglie equation, which describes the wave properties of matter and says that an electron can have wave nature also, described by lambda equal to h by mv. So if we include the mass of the electron, which is around 9.1 into 10 to the power minus 31 kg, and we include the velocity of an electron, we will find the wavelength of the electron to be much smaller compared to the wavelength of light. So an electron microscope having much smaller wavelength, then diffraction will not happen. Instead of diffraction, it will simply, so diffraction when it happens, when you have slits like this and the light is passing through the slit and getting split up. But if the wavelength of the light is so small that it can just pass through this grating without having any diffraction, then you will have a sharp image. That is what electron microscope does. Basically, if you put the value of h, mass of an electron, and velocity of an electron, then you will see that the wavelength of electron is much smaller than the wavelength of light. And so there will be no diffraction in case of an electron, and you will be able to get a sharp image. Also, because the wavelength of an electron is much smaller, the resolution is much bigger. So resolution we saw is lambda by 2 Na. So smaller wavelength will give higher resolution. You can see electron microscope can be used to even look at virus, DNA, sugar molecules, or even up to atomic resolution, whereas light molecules can only observe up to certain bacteria. So the resolution power is also greater for the electron microscope. In electron microscope, basically, instead of a beam of light, a beam of accelerated electrons is used as a source of illumination. Electron microscopes use shaped magnetic field to form electron optical lens system that are analogous to the glass lens of an optical light microscope. Let us look into the two types of electron microscopy. First is the transmission electron microscopy, or TEM. In this, we look at the setup. So first, an electron beam is produced by an electron gun commonly fitted with a tungsten filament cathode as the electron source. So this tungsten filament cathode can be heated to cause thermionic emission of electrons. The electrons that are emitted is accelerated by an anode. So you have an anode with a positive charge. So the negatively charged electrons emitted will be accelerated towards the positively charged anode, typically at a potential difference of 100 kiloelectron volts with respect to the cathode. And then you have electrostatic and electromagnetic lenses. So what this is, in case of light, you had glass lenses where when the light was passing through these lenses, they were getting refracted at different angles. Here what you have is a force field. Basically, electrons are passing through this force field, and you have set up an electromagnetic force field here. And you know, electromagnetic force field has both electric and magnetic components. So it will interact with the negatively charged electron and it will focus the electron. You can see the electron is passing as a beam, but when it passes through this lens, it is getting focused at a particular point. So this is exactly how, when light rays pass through a glass lens, they get focused at their focal point. That is exactly what this condenser lens is doing. There is an electromagnetic field in the condenser lens, which is focusing the electron beam on the specimen. Now, this is the specimen or sample. After the electron beam emerges from the sample, it carries information about the structure of the sample that is magnified by the objective lens system. So now this electron beam that is coming out, it has information about the structure of the sample. But here the information is still too small to observe. So it has to be magnified by this objective lens system. The spatial variation in this image may be viewed by projecting the magnified electron image onto a fluorescent viewing screen. So what this means, if we again look back at the light microscope. So in light microscope, what was happening? The light was passing through the objective lens and forming a real image. And then the light was passing through the eyepiece lens and forming a hugely magnified virtual image. The concept here is the same. You have the electron beam passing through the sample going to the objective lens, and then it passes through two more lenses, the aperture lens and the projection lens. And finally it gets, so these are electron beams. You cannot observe them by the naked eye. So what it is done, a fluorescent screen is taken where this beam of electron is allowed to hit. So wherever the electron is hitting, an image is forming. You can think of it like a photographic film. So in photographic film, when you expose it to light, wherever the light is striking, there will be a dark spot forming. Similarly, in this fluorescent screen, wherever the electron beam is hitting, dark spots will form, and that will give you an idea about the structure of this sample. So this is basically like a compound microscope, except instead of glass lens, you have electromagnetic fields which are focusing the electron beam. Firstly, they are focusing it on the sample, and then after passing through the sample, they are magnifying the beam so that it is visible to the naked eye when it touches this fluorescent screen. From the image developed on this fluorescent screen, we can have an idea about the structure of this sample. Now, this is called transmission electron microscopy because the electron beam is being transmitted through the sample. Now, the resolution of this transmission electron microscopy, or TEM, is limited primarily by spherical aberration. If we look back, spherical aberration was basically when the beam of light striking at the edges of the lens was being refracted to a different extent compared to the beam of light striking in the middle of the lens. So here also same thing happens. the beam of light that is passing through, here only a small region is shown, but it actually passes through the entire lens. So when the beam of light passes through the edges of the lens, that is being refracted to a different extent compared to the beam of light passing, beam of electrons passing through the center. So this kind of spherical aberration occurs in TEM also, and that will cause a loss in resolution. So this can be solved by using something called a high resolution transmission electron microscope, or HRTEM, and that will help you resolve to below 0.5 angstrom, which is a huge resolution, like a resolution of the atomic level. You can observe individual atoms by this resolution. So the magnification is more than 50 million times, which is incredible if you think that the initial light microscopes could only magnify by about 10 times. And now we have come up with a transmission electron microscope, which can magnify more than 50 million times. So that is a good point of the TEM, that it can cause such high magnification. The disadvantage is that you need very thin specimen, around 100 nanometers. So the specimen that is kept here, the electron has to pass through the specimen. That can only happen if the thickness of the specimen is very less, around 100 nanometers. If the specimen is thicker than that, then the electron beam will simply be absorbed and reflected, or it will not pass through the specimen. So you will not get an idea about the structure of the specimen. So that is one disadvantage, is that this sample slice has to be very thin for TEM. The second kind of electron microscopy is called SEM, or scanning electron microscopy. Here what happens, I will again go through the instrumentation. Electrons are produced at the top of the column, and they are accelerated down and pass through a combination of lenses and apertures to produce a focused beam of electrons which hits the surface of the sample. So here the beam does not pass through the sample, it is just focused on the surface of the sample. The position of this electron beam on the sample is controlled by scan coils situated above the objective lens. So there is something called a scanning coil here that is fixing the position where the beam is hitting the sample. So by changing the scanning coils, we can allow the beam to hit at different regions of the sample. So sometimes they will hit here, then it will hit here. So basically these coils, by controlling the coil, we can allow the beam to be scanned over the surface of the sample. So the beam will scan over the entire sample surface, and it collects information about a defined area on the sample surface. Now, as the electrons are interacting with the sample, they produce secondary electrons. That is, they are hitting the sample and they can excite one of the electrons on the sample to move out, or they can produce backscattered electrons. So some of the electrons can hit the sample and scatter back. And characteristic X-ray. Characteristic X-ray means that these electrons are hitting the sample. It is pushing out one of the inner electrons of the sample, and the outer electron of the sample is jumping down to take the place of the inner electron. So that will cause emission of a certain X-ray called characteristic X-ray. So all these signals are collected. When the electron beam hits the sample surface, you can have some electrons scattered back, that is being collected. You can have the sample lose some of its own electrons, which are called secondary electrons, or this electron beam can excite one of the inner electrons of the sample and push it out of the atom, in which case the outer electron of the sample will jump down to take its place and a characteristic X-ray will be formed. So all these kinds of X-rays and electrons are collected by the detectors to form images which are then displayed on the computer screen. So in SEM, what happens? In TEM, we saw the electron beam is passing through the sample, but in SEM, the electron beam has just been focused. In SEM, the electron beam is being focused at one particular point in the sample. And by controlling the scanning coils, we can change the point at which the electron beam is hitting the sample. So suppose you have a certain crystal and the electron beam is scanning over the entire surface of the crystal. While the electron beam is scanning over the crystal surface, it is forming different scattered electrons or secondary electrons or X-rays, all of which the detector is picking up. And when passed through a computer, the computer will form an image of the sample surface. So SEM gives a very good idea about the structure on a sample surface. Typically, modern full-sized SEMs form resolution between 1 to 20 nanometer, whereas if they are connected to a computer system, the resolution can be 20 nanometer or more. So TEM actually produced higher magnification, around 0.5 angstrom. But the problem with TEM was that you needed a very thin slice of the sample so that the electron beam can pass through the sample. And SEM, you don't need a thin slice of the sample because it is hitting the sample surface, not passing through the sample. So the resolution in SEM is lower, but at the same time, you don't need to slice the sample very thinly. So that is an advantage of SEM over TEM. Now, there are certain challenges when you are studying the structure of a biological material. It was noted that electron microscopy unfortunately could not be used to study biological material without the destruction of the organic cells by the intense electronic bombardment. So then there is no point, right? If you're observing the cell, but the cell itself is dying, then it is not a very good method. So this required scientists to come up with a new method, which basically led to cryo-electron microscopy. Another major problem was how to preserve water in the biological sample in the vacuum maintained inside the electron microscope chamber. So in the electron microscope chamber, there is a vacuum because if there is no vacuum, then the electrons will hit the air particles and generate ions and cause a lot of problems. So vacuum needs to be maintained in the electron microscopy chamber. But the problem is that suppose here you have a protein which is being stabilized by a thin layer of water. Now, while you are creating vacuum, the water is evaporating. The structure of the protein will also collapse due to surface tension. So, you needed a method by which the water does not evaporate in the vacuum. So, again, this was something that led to cryo-electron microscopy. And another problem was that intact biological material has very low image contrast as most high energy electrons pass through the specimen. So if the electron is passing through the specimen, you will not get a very good resolution. You will only get a resolution if the electron is interacting with the specimen atoms. So what was the strategy for solving these problems? Firstly, it was suggested to cool the biological material so that it does not get destroyed by this electronic bombardment. And another is that the probability for multiple electron scattering events must be negligible. So the sample must be thin, ideally composed of only a single layer of the particle of interest. So if the sample is thick, there will be several electron scattering events. And again, the resolution will become very poor. So for that, the sample needs to be very thin. This was what led to cryo-electron microscopy. So we'll go into that slowly. Now, to perform single particle electron microscopy, thousands of identical particles are imaged and computationally averaged together. The ideal single particle specimen should be thus as homogeneous as possible, because heterogeneity can result from various ways. There can be a variation in conformation or flexibility, or the composition itself can change. So when you have a protein, the protein is a dynamic molecule. It can move about, it has a certain flexibility, it has a certain variation. So that will cause the protein to be heterogeneous. Also, compositional changes means suppose the protein is interacting with a ligand that will change the composition and cause heterogeneity. But this heterogeneity is a problem because what electron microscopy does is that it will image thousands of particles and then take an average. So if all the particles are heterogeneous, you will not get a very good image. You will only get a good image if all the particles are homogeneous. So the first thing to be done was to reduce this conformational variation. Conformational variation can be reduced biochemically using appropriate protein purification methods. So the first thing was obtaining a very pure protein so that impurities can be removed and that heterogeneity due to the impurities can be minimized. However, just ensuring that a sample is biochemically pure does not ensure that the sample will appear homogeneous. So in that case, what needs to be done is that either you can change the solution buffer conditions. So this is a kind of trial error method where you look at the sample in the microscope and you change the buffer conditions to such a place where the microscope gives the clearest image. So in that way, the buffer condition can be optimized to maximize the resolution. Another technique is chemical cross-linking. So suppose you have two different kind of particles, this red particle and this blue particle. So if you link them by a cross-linker, then your particles will become homogeneous because instead of having two different kinds of particles, you now have a single kind of particle which is made up of one red strand and one blue strand. So this is bringing a homogeneity in the system. But this can also have some drawbacks. Chemical cross-linking may introduce artifacts such as trapping the complex in non-native or non-functional form. So if by cross-linking you somehow change the structure of this blue or red particle, then that is not ideal because you want the blue and red strand to be in their native conditions. You just want to join them by a linker so that they are homogeneous. But if joining them by the linker is destroying the native structure, then that is not ideal. Another approach to reducing conformational flexibility includes ligand-induced stabilization, where molecules such as a substrate, ligand inhibitor, or protein nucleic binding partners. So basically what they are saying is that you have a protein and you are attaching a ligand to the protein. So the protein, which was initially very flexible and it used to move around a lot, by attaching the ligand, you are minimizing the movement of the protein. So if the movement of the protein is minimized, then the conformational homogeneity is increased. So this is another way in which you can reduce the dynamism of the system to get a clearer image by electron microscope. Now we will talk about the sample preparation. So to image with electrons, a microscope is maintained under a high vacuum in which hydrated biological specimens would dehydrate rapidly. We just saw that in high vacuum, the water around the biological specimen will be evaporated and the sample will just collapse. So to image a biological specimen, it must be fixed, preferably in a native state. Another consideration is the sample thickness. We have seen if the sample is too thick, then there will be a lot of scattering of electrons and the electron will not pass through the sample properly. So in TEM, transmission electron microscopy, one prerequisite is that the sample must be thin enough for the electron to pass through it. So the specimen needs to be processed to make it thin enough. We saw the limit was around 100 nanometers so that the electron can pass through the sample. Now the sample is mounted on a grid. Electron microscopy grids are traditionally 3.05 millimeter across and made from a mesh of metal. Copper grids are most commonly used, but gold supports are becoming more common, especially when cells are to be grown directly on the grid because gold is non-toxic to cells. This metal mesh supports the sample film on top. So over the metal support, a support film is deposited. So you have the metal grid. On top of the metal grid, a support film is deposited. Finer mesh sizes provide more support for the film, but when the grid is tilted, the bar can block the beam. So it has been found that the optimum mesh grid is a 200 mesh grid for data collection. Now, on top of this metal grid, you have the support film, which is generally a carbon film with regular holes where the sample can be placed. And in each of these holes, you can see the sample is placed. And the sample is basically prepared in a way we will see in the next slide. So negative stain is a common method for examining a specimen, especially macromolecular complexes at room temperature with high speed. Typically, specimens are adsorbed onto a thin, around 10 nanometer continuous carbon support film. Then it is stained with a solution of heavy metal salt, commonly 1 to 2% uranyl acetate or uranyl formate, which generates a cast around the object. So you have the cell which is stained by this heavy metal salt, which is forming a kind of layer around the object. This layer scatters electrons more strongly than the encapsulated material and thus generates a relatively high signal to noise ratio. So why this layer of the stain is essential is the original cell is not scattering electron very strongly. So the signal to noise ratio is low, but the stain layer around the cell is scattering electrons very nicely. So you are getting a better image of the cell. Also, this layer is more resistant to electron damage and prevents collapse of biological material during drying in the vacuum within the electron microscope. So electrons, we saw that in a normal electron microscope, they can hit the cell and destroy the cell. But if you have this layer of stain protecting the cell, that will not happen. Also, we saw that in the vacuum that is there in an electron microscope, the cell structure can completely collapse due to evaporation of water. But this stain layer will protect that cell structure from collapsing. So this is called negative stain, and it protects the cell from damage by the electron microscope and also gives a better signal to noise ratio image of the cell. Deformation of the specimen during adsorb. Now, what is a negative thing that can happen is that when the stain is being adsorbed on the surface of the cell itself gets deformed. That is a negative thing because you want to find out the native structure of the cell. If it is getting deformed by the stain, then that is a bad thing. Additionally, structures limited to modest resolution greater than 20 angstrom is possible by this method because the stain is sitting on the surface of the cell. So because the stain is also formed of different granules, you cannot get resolution greater than 20 angstrom because the stain granules will prevent greater resolution in the microscope. So that is another negative point of the stain. And another thing is that the stain is incompatible with certain buffers and reagent. So suppose your protein or cell is stable only in that buffer and the stain is incompatible with that buffer, then you cannot use this method. And we saw that we used a carbon film, but sometimes what can happen is that when you're using a carbon film, the sample will attach to the film in such a way and change the native orientation of the sample. So that is also a negative thing. Basically, you want the carbon film to support the sample and the stain to help the visualization of the sample, but the native structure of the sample should not be affected. So that is a very important thing during any of these visualizations, that the native structure of the cell should not be affected in any way. Just give me 1 minute. Okay, now we will look at this sample preparation technique. The sample preparation technique involves the dehydration of the biological specimen, which will remove them from the native aqueous environment. So removing the water is a bad thing because we saw when water is removed, the structure collapses. So what is done is cryo-immobilization, which will preserve that aqueous environment while also ameliorating radiation damage. What this means is that cryoimmobilization will maintain the water environment so that the cell structure is not destroyed, and also because you are cooling the cell, it will minimize the damage due to the electrons striking the sample. So how this is done, the sample must be frozen very rapidly at a rate of around 10 to the power 6 degrees Celsius per second, so that the water in and surrounding the specimen is fixed in a vitreous state. That means whatever the state, imagine this is a cell with water around it. If you freeze it at this fast of a rate, 10 to the power 6 degrees Celsius per second, then the water will freeze in the orientation such that the cell structure or the protein structure is maintained. Otherwise, if the freezing is happening very slowly, this kind of ice crystal will be formed and the cell structure will collapse. Or if the specimen is warmed above minus, so this cooling is happening at minus 196 degrees. If somehow the temperature goes up to minus 137 degrees, again this crystal ice will be formed, which will destroy the 3D structure. So you want a very rapid rate of cooling, 10 to the power 6 degrees Celsius, and very low temperature of minus 196. Only then the water will freeze in the native structure of the cell, and the cell structure will be maintained. Ice contamination compromises the structural integrity of the specimen, as crystals withdraw water molecules from hydration shells of the specimen or the specimen itself, and it makes the structure of the cell collapse. Also, these ice crystals will degrade image quality because they diffract electrons. So what is needed is very rapid freezing at very low temperatures. Again, another thing is when samples thinner than the edge of a large cell can be vitrified. That vitrified means freezing the water in the native orientation. So samples thinner than the edge of a large cell may be vitrified by plunge freezing. Thicker samples of less than 1 micrometer, such as the nuclear and perinuclear regions of eukaryotic cells or tissue sections, are likely to experience some crystalline ice formation during plunge freezing. So this technique that you saw, very rapid freezing at low temperatures called plunge freezing, it will work for thin segments. But if the specimen is thick, like the nuclear or perinuclear regions, then even during plunge freezing, there will be some crystalline ice formation. And you can see, so here you have a thick section of the cell and you are carrying out plunge freezing at the normal temperature, pressure or ambient pressure. You can see this kind of hexagonal crystalline ice is forming, which is breaking the cell membrane and destroying the structure of the cell. So the solution to this is high pressure freezing or HPF, which involves raising the pressure of the sample to around 2000 bars while dropping the temperature using liquid nitrogen. So you do the freezing to minus 196 degrees, but additionally you add high pressure of around 2000 bars. So that will maintain the structure of the cell. It will not allow the ice to form the hexagonal crystals, and the structure of the cell will be maintained. So the advantage is that the sample is preserved in a native-like state and you can get high resolution 3D structural information. The disadvantage is that to be visualized by transmission electron microscopy, the specimen needs to be very thin. But in this case, the specimen is not thin enough to be observed by TEM. So somehow the specimen needs to be made thinner by focusing an ion beam after freezing. So this is basically to generate this kind of focused ion beam, to study thick samples, it is a bit technically challenging. That is, the instrumentation required is more complex and costly. So this is how high pressure freezing, along with the plunge freezing, helps to study thicker samples. Now we'll talk about the electron source. The electron gun of an electron microscope extracts and accelerates electrons and is typically either a thermionic electron source or a field-assisted thermionic emitter, such as a Schottky emission gun, commonly known as a field emission gun. What this means is that the source of the electron is either thermionic emission. That is, suppose there is a tungsten filament, you are heating the tungsten filament and the electrons are being ejected. That is called thermionic electron source. And the other thing that is called field assisted thermionic emission or field emission gun. Basically, again, you have a tungsten tip which is being heated and you are getting the electrons, but additionally there is a potential difference developed here, that is a field that is helping in the emission of the electrons. So this is a thermionic emission source where only by heating the electrons are being emitted, and this is a field emission source where the emission is happening because there is so the electron is negative and the anodes are positive. So the positive charge from the anode is pulling the electrons away from the tungsten. So this is called a field emission source or field emission gun or FEG. So the common thermionic sources can include tungsten filaments or lanthanum hexaboride crystals, that is LaB6. And they are heated so that the voltage potential exceeds work function. That is, the voltage is, they are heated to such an extent that the electrons have enough energy to go free from the filament, and they operate at voltages around 80 to 200 kilovolts. In comparison, the field emission gun, basically what happens, there you have instead of a broad tungsten filament, you have a very thin tungsten filament coated with zirconium oxide, and it operates at much higher voltages of 200 to 300 kilovolts. Here, the voltage is what is pulling the electron away from the tungsten tip, so the voltage needs to be higher. FEGs are much brighter and more coherent compared to the conventional thermionic sources, so the field emission is preferred. But the problem is the field emission is also very expensive. So depending on what you need, either the thermionic emission source is used or the field emission source is used. Now we will look at the detectors. Cryo-EM images of biological specimens are intrinsically noisy due to low electron doses used to prevent specimen radiation damage. If you use too high electron radiation, the specimen will be damaged. So only lower radiation can be used, and so the signal to noise ratio is not great. A perfect detector would add no noise, but in practice, all detectors have some level of noise. And this is expressed as detective quantum efficiency, DQE, of the detector, which is the square of the output signal to noise ratio over the input signal to noise ratio. So the detective quantum efficiency of any detector, that is DQE, in the numerator you have the signal to noise ratio of the output, and in the denominator you have signal to noise ratio of the input, and both are squared. The detector generally used is a charged coupled device or CCD cameras, which utilize a phosphor or similar scintillator, which induces the emission of photons when electrons strike it. So basically what happens, you have electrons, and when the electrons strike the pass through the sample and they hit the detector, in the detector photons are produced, and these photons are transformed into electrical signals, and finally an image is obtained. So you can have in charge coupled devices, you can have two kinds of detector. Firstly, you have the indirect detector, where the electron passing through the sample hits the detector, a photon is produced, and the photon passes through fiber optic cables to give the image. This is the more primitive detector. The modern detectors are CCD, or charge coupled detectors, where you have a phosphor or other scintillator, where so the electron is striking and creating photons. That much is the same as the indirect detector. But in the indirect detector, the photons directly form the image. Here what happens is that the photons are converted into an electrical signal. And then there are many photosensitive elements where this electron signal passes through, and it is converted into a voltage, and it is read by a detector. So this is called the charge coupled device. So the photons which are converted to an electrical signal, it is read out and amplified and digitized to form an image. So this is the more modern kind of detector. In the previous detectors, the electron would just strike the detector and photons would be formed, from which you will get an image directly. In CCD, these photons are converted to an electron signal, which is then read by the detector and amplified to form an image. Now about the data collection. Once the protein density and ice thickness or quality have been optimized, an initial data collection should be performed on a mid-range electron microscope. The goal is to obtain 2D reference-free class averages which have high resolution features at the level of secondary structure, that is alpha helices or better. So what this means is that you are collecting the data. What the data will give you is, it will give you different 2D pictures of the 3D sample. So if you have a 3D sample and you can imagine taking the cross section of the sample from different angles. So that cross section is a 2D image or 2D projection of the sample. So you will get a set of 2D projections in the initial data collection. These 2D projection will have high resolution features at the level of secondary structure like alpha helices. Once this preliminary data collection is done that generates suitable 2D classes, a larger data set with a few hundred micrographs and several tens of thousands of particles is collected on a mid-range or high-end microscope, which forms the initial 3D map. So again, I'll explain. This initial data collection is to find out that what are the different kinds of 2D projection you can get. So basically to find out the different 2D classes. Once you have the 2D classification, you can generate several other, like few hundred to tens of thousands of other 2D images on a better resolution microscope. So you can see here it is 2D classification. So this is one projection image. You are collecting hundreds of this projection 1, hundreds of projection 2. So several thousands of 2D projection data you are collecting in a mid-range to high-end microscope. And finally, all these 2D projections are put together to form a 3D map of the structure. So and this 3D map can then be refined to build a model. There are certain important factors that need to be kept in mind. Firstly, do the 2D classes show distinct views of the particle with self-consistent dimensions, each with secondary structural features? So you see this 3D classification. So we need to, in order to reconstruct a three-dimensional structure, we need two-dimensional images from different angles, like from the top, the side, from the bottom. We need several distinct views of the particle. And each of the 2D projections must have high enough resolution that we can see the secondary structure with it. The second point is that, is the orientation distribution sufficient to allow calculation of a 3D structure with isotropic resolution? So sufficient, the 2D projections must be obtained from different orientations to properly reconstruct the 3D structure. If the orientation distribution is not suitable, one can alter the buffer, add detergents, change the conditions, or use an alternate film surface to get a better picture of all the different orientations. And if the resolution of the initial 3D reconstruction reaches a reasonable resolution for the number of particles and image acquisition settings, then you can collect even more data on a high-end microscope to increase the resolution. That is basically refinement. So the initial 3D structure can be refined further and further by getting more images of the 2D projections. So the initial 3D reconstructed structure can be refined to improve the resolution. Now, every data set collected should be verified that the specimen preparation and illumination conditions are optimized and that the conformational variability is minimized. So we had seen that if there is a variation in conformation, that is dynamics is there, then resolution would be decreased. So that should be taken care of, and also the illumination and everything would be maximized to get a well-resolved image. Currently, the simplest way to do that is using a per micrograph motion correction program like Motion Chord. So this is a software that you can use, Motion Chord. It will optimize each projection so that the final image obtained is of the highest resolution. So each micrograph collected, that is each projection collected, you can imagine it as a movie subdivided into individual frames. What the software will do, it will ensure that the dynamism of the particle is minimized, and it will kind of calculate and recalculate to optimize both the image, and the dynamism of the particle will be minimized to give the best resolution. If there is a large amount of specimen movement, that can be detected using real-time Fourier transform of the collected micrographs. So if a large amount of movement is detected by this Fourier transform, then different techniques have to be used, like we studied earlier, like cross-linking or cryogenic freezing to minimize the particle movement. So basically that is the, you can see here it is, or sometimes the grid can be changed. You can see that if the grid is completely gold, the movement is very low. So here on the x-axis you have movement. If it is being done on a gold film, you can see the displacement is only like 2 angstrom. But if you are doing it on a carbon film, there is a large displacement from around 5 to 35 angstrom. So then the gold film is preferred, which has a very small displacement of the particle. So by changing the film also, the resolution can be optimized. Then about the data processing. So the single particle extracted from the micrograph are sorted and filtered using 2D classification methods. So you can find out that which of your images will fall under. So you can see this is a 3D structure and these are different 2D projections of the structure. So each image that you get can be classified into a different 2D projection, and you will get several identical copies of the protein complex in different orientations. All these 2D projections are put together and combined to generate the 3D structure. So the 3D structure can be randomly oriented in solution, and depending on the orientation of the 3D structure, you will get different 2D structures. And using the 2D structures, you can reconstruct. You can see these are different 2D structures. They are reconstructed to get the 3D image. So this is all part of the data processing and analysis. And before processing to the reconstruction, it is necessary to know in which direction the particles are facing. So you have, see, these black squares are different 2D projections. it is important to know in which direction the particle is facing in each of these projections in order to reconstruct the final 2D, 3D image. So this orientation can be determined using Euler angles. They can be defined in two ways. In the first definition, the orientation can be determined by using latitude, longitude, and rotation. So suppose I am looking at this particle, this projection. I have to find out so if you imagine it as a sphere, so latitude and longitude will give you an idea about which region the 2D projection is coming from, and and also the rotation of the particle. So as you are rotating, so this from the top, if you look at the particle, you will get an image like this. If you look at the particle from the side here, you will get an image like this. So that is the rotation. So if you have an idea of the rotation, that is from which direction you are looking at the particle, and you have an idea of the latitude and longitude, then you will find out that the 2D projection is coming from which region of the 3D structure. So if you have that information for all the 2D projections, you can reconstruct the 3D structure. Another way is that the initial position is determined using the z-axis. So z-axis is maintained at z, and the particle is rotated from initial, it was x1, the initial x-axis was x1. The particle is rotated around the z-axis so that the xy planes are changed. So for the initial xyz axis, you will have a certain projection, and when you rotate the z-axis, you will have another projection for the new x and y axis. So these projections, if they are put together, they will give you an idea around which axis the particle is rotating, and that will help you reconstruct the three-dimensional image. Same thing can be done for, you can see this is rotation along the x-axis. So like this, by rotation along the different axes, you can find out the positional orientation of the 2D projection. So for the 2D projections, if you can find out that from which region of the 3D structure the projection is coming from, and from which latitude and longitude make up this. So like this, you can see, if you look along z-axis, you will get this projection. So if you can find out the XYZ coordinates also, it will help you put all the 2D projections together to reconstruct the 3D image. The goal of this 3D reconstruction in cryo-electron microscopy is to take a 2D projection and turn it into its 3D volume. So this will allow for the visualization of small structures within the sample. It is essential to reconstruct the 2D projection because the information gained from the projection itself is limited. Each point in the 2D plane corresponds to a slice in the 3D volume. It is possible to obtain a 3D volume only if we put together all the 2D projections and take the Fourier transform of the projections to reconstruct the 3D volume. So you can see these are different 2D projections. You can put together the different projections and do an inverse Fourier transform to get the 3D structure. So again, I will explain this part, just one second. So a molecule can have many different orientations. For different orientations, you can get different 2D projections. And the 2D, from the 2D projections, you can have a 2D Fourier transform. These 2D Fourier transforms are put together and an inverse Fourier transform is done to get the reconstructed 3D structure. And from that, you can get the three-dimensional model. And you can refine the model further by taking more 2D projections to get a better resolution. These 3D images of the biological sample is obtained by a process called cryo-electron tomography, where a 3D reconstruction of sample is created from 2D tilted images. So you can see there are so many 2D images. They are kind of put one after the other in a tilted manner. And using cryo-electron tomography, you can reconstruct the 3D image. Raw images from different projections are recorded and they are stitched together using computer software to form the required 3D image. Then image enhancement is done, where image noise is the random variation of brightness or color information in images produced by the sensor. During image enhancement, this noise is reduced so that you can see, compared to this where there is a lot of noise, in this structure, the structures are much more clear. There is a lot of contrast and the outlines are very sharp. So this is called image enhancement, where the structure resolution is improved. So you can see the resolution progression of cryo-electron microscopy. It is represented by the resolution of this molecule glutamate dehydrogenase. Initially around 2003, the resolution was greater than 4 nanometer. And at 2017, the resolution has gone up to 0.36 nanometer. So over the years, as the technology has improved, the softwares have improved, the resolution has also greatly improved. And unlike X-ray crystallography, which locks the crystal protein into a single static pose, researcher can use cryo-electron microscopy to calculate the structure of a protein that has been flash frozen in several conformations, and so it can deduce the mechanism by which it works. So what it means is that in X-ray crystallography, you can get only a single conformation of the protein. But when you have the protein solution and you do plunge freezing to do cryo-electron microscopy, in that frozen sample, the protein will occupy many different orientations. So like you saw, the protein will occupy many different orientations. So each of the conformations can be determined using cryo-electron microscopy. And so if you have many different conformations of the protein, it can be used to deduce the mechanism of the protein function. So that is another advantage of cryo-electron microscopy over X-ray crystallography. And there is something called a protein data bank. One second. So there is something called a, hello? Did someone say something? There is something called a protein data bank for three-dimensional structure of large biological macromolecules. So the protein structure determined by X-ray crystallography or NMR spectroscopy or cryo-electron microscopy, they are submitted in this protein data bank and they can be freely accessible by any researcher around the world. So in this, when someone is depositing a structure in the PDB, all the experimental information is also included as to how the structure has been obtained. And currently, this is the only repository for 3D biological protein structures solved by different experimental techniques. So in the protein data bank, there are certain tools. One is for data extraction. So you have this command called PDB extract, which can extract and download the data in a PDB format so that you can open the PDB using a software like Discovery Studio to look at the structure. Then you have a tool called SF tool, which will convert the structure factor file in the various formats. If you remember, during X-ray crystallography, we discussed structure factor, which is something which determines the orientation of the molecule in the crystal. So using this tool, you can convert the structure factor file into the format you need. There is also a dictionary called Ligand Expo, which will give you the IDs of different ligands. So if you have a protein, along with that protein, there is a series of ligands which can bind with the protein. and all that information is also deposited in the PDB. And there is another command called point suit, where you can have symmetry records for the different macromolecular assemblies. So suppose you have a PDB file deposited where there is a protein interacting with another protein, which is basically called macromolecular assembly. So this command point suit will give you the different symmetry operators that are valid on that assembly. Now, when you are depositing any structure, it has to be, before it is accepted into the protein data bank, it has to be validated. So what you do, you submit a structure to the protein data bank, and the software will check the quality of the structure, and it will, if there is any problem with the structure, it will highlight those problems. So suppose there are Ramachandran outliers. What does that mean? We saw in Ramachandran map, there is a allowed and disallowed region of the amino acid. So suppose in the protein structure you have given, many of the amino acids are in the disallowed structure. Then the validation report will tell you that this cannot be a valid structure because there are many outliers. Similarly, there are other factors which find out the validity of the protein structure you have deposited, and if it passes the validity report, only then it will be accepted into the protein data bank. So ultimately, this validation will give you a summary of the quality of the model compared with other deposited models in the PDB to help you assess the quality. So if compared to other models in the PDB, the model you are depositing is having a lot of negative points, then the validation report will say that these points need to be improved before the structure can be accepted in the PDB. There are many 3D structure viewers. Yes.
Ma'am, in the previous slide, there is 14.5%, 85 value. These are all the values. So, in case of Ramachandran plot, it must be greater. The plot means the protein must be greater than 90%. Then it is called, it is a better protein. Means we can
Yeah.
take that protein structure.
Yes.
But ma'am, in the case of this value, this is Ramachandran outliers. Means this 14.5% is the outlier.
Outlier, yeah. So this is not a very good protein structure because 14.5% of the amino acids is lying in the disallowed region of the Ramachandran.
So I think, but 10, if 10%, if 10 or less than 10, it would be, then
Correct.
it would be a good
Correct.
protein.
Correct. This is
Okay.
just an example shown where the report is not very good. So this kind of structure will not be accepted in the PDB.
So, ma'am, why we are using this means we can, in spite of doing Ramachandran plot, we can use this because I think this is present in the PDB structure. This means this image is present in the PDB structure when we search for any protein. So, so then why we are doing Ramachandran plot means we can also take the values from here and say that the protein structure is good or not.
That is only done. I am not understanding your question. Like Ramachandran plot, we don't physically do the Ramachandran plot. Like in this program, which is calculating the validity report, the Ramachandran plot is already, like the program is checking for it. We don't have to separately plot it.
No, ma'am, but in case of some proteins, like if we are driving, if you are taking the protein structure, means in the FASTA format or PDB format, then again we are putting that sequence of protein in the Ramachandran plot, means to calculate the Ramachandran plot in certain softwares. So why we are doing that? If we are already having the value, then why we are doing that kind of thing?
I don't know, like I have never done that. Like that is maybe people just do it to check, but there is no application of doing that.
So in our case means in bioinformatics we check that. in for the Ramchandran plot. So I was asking.
I am also
Okay.
not sure actually why that is done.
Okay,
So
ma'am, means
yeah.
there is no difference if we calculate the
There is,
Ramchandran
it's the
plot.
same thing. It's the
Uh,
same
uh,
thing. You can do it from here also. You can do it separately also. So yeah.
okay, okay, ma'am.
So you have many 3D structured viewers where if you include the PDB ID, you can download the PDB format and you can look at the structure in a three-dimensional way. So there are certain views. So there are, you can see, there are certain softwares which will help you look. So here the amino acid sequence is given, and above that it will give you the secondary structure. So this region is forming a beta sheet. This is a turn. This is a helix. So all this information will be deposited in the PDB file, that which region of the amino acid chain is forming what secondary structure. And also you can have correspondence between the PDB sequence and genome. So you know from triplet coding that three base pairs will code for one amino acid. So from the amino acid sequence, you can find out what is the DNA sequence, that is the genome sequence. So that information can also be found out from the triplet coding, where three base pairs are coding for one amino acid. So there is a protein featured view where you can see the amino acid sequence and secondary structures, and there is a genome view which will tell you that this particular amino acid region comes from what DNA sequence. So now this in the more recent PDB structures, this genome view is also included. So you can do that. You can correlate that from which region of the DNA this particular protein is being formed. Now, as you have gone through the years, there has been a large exponential growth in the number of structures deposited in PDB. And you can see that the number of cryo-electron structures are also increasing. As so, this orange is the number of cryo-electron structures that has started increasing more recently, like around 2006 or so, because cryo-electron microscopy is a relatively new technique compared to X-ray or NMR. So if you compared with X-ray and NMR, the number of X-ray structures is still much greater. Cryo-electron microscopy structures is still very low. But as the years are going, more and more of the cryo-electron structures are being deposited, because that is a more modern technique. And as time goes, you will see that the number of cryo-electron structures will gradually overtake the number of X-ray structures. So cryo-electron microscopy enables the determination of 3D structures of macromolecular complexes and cells from 2 to 200 angstrom resolution. So 2 angstrom resolution is a very high resolution that can be obtained by cryo-electron microscopy. And there is a database called EM Data Resource. Basically, that database, what it does, it establishes data validation methods that can be used in the process of structure determination. So like we saw in the case of Ramachandran outliers and other points, there are various criteria that the data validation methods give you, that if those criteria are fulfilled, then the determined structure is a good structure. It defines key indicators of a well-determined structure that should accompany every deposition. So whenever a structure is being deposited, what are the main features of the structures that need to be deposited? That is also mentioned in this EM data resource. And finally, appropriate validation techniques are included so that the software that is checking whether the structure is a valid structure or not, that software can function very fast. So EM data resource is basically available in order to quickly validate the structure that is being deposited in the PDB. And we'll talk about certain data banks. So we saw the protein data bank. Another is the nucleic acid data bank. it is a portal for three-dimensional structural information about nucleic acids. So whatever is the structure determined for different nucleic acids or complexes of nucleic acids, that is being deposited here. So you can see 42% of it is basically protein DNA. That is DNA that is coding for different proteins. Lesser percentage is for protein RNA. So RNA also codes for proteins in several viruses. So similar to protein data bank, this nucleic acid data bank is there. And you can see initially most of the structures were determined by X-ray. But as the years are going, now you can see in 2021, the cryo-electron microscopy, this red bar, has increased a lot. So gradually now more and more structures are being determined using cryo-electron microscopy, and X-ray is still present, but slowly cryo-electron microscopy is overtaking it. And people, like they use the protein data bank, they can use the nucleic acid data bank to perform searches based on this code that defines each structure, and they can download and observe the structure to learn more about it. Then you have the Biological Magnetic Resonance Data Bank, or BMRB. This is a repository for data from NMR spectroscopy on different proteins, peptides, nucleic acids, and other molecules. So for any protein or peptide or other biomolecule, it will give you the 1H spectra, it will give you the 13C spectra, and it will also give you different 2D spectra and everything. So this is another depository which gives you very important spectral and quantitative data about NMR. So whatever molecules have been studied by NMR, they have been deposited in this biological magnetic resonance data bank. So suppose you have an unknown NMR spectra and you are predicting it belongs to a particular protein. You can search in this data bank for the reported spectra of that protein, and you can compare, and that will help you find out whether the unknown molecule you have is that particular protein or not. So this data bank also it is available to all researchers, and it helps people understand and analyze the structure, dynamics, and chemistry of biological systems, and it provides information to researchers to further improve the field of this NMR spectroscopy in biomolecules. So we have
you said about 1H spectra and other spectra.
yes,
What are these spectra?
So I had a class on NMR. Did you attend that? I think it was week 6. So this is week 8. There was a week 6 class where I discussed about all NMR, like 1D, 2D, 3D, all the NMRs I discussed. You can look into the videos I have posted. So I think that will help.
Okay.
So I'll go into some of the questions now. So it is, first question is asking, what is the resolving power of the unaided human eye?
0.25 micrometer. Oh, sorry, millimeter.
Yeah, millimeters. So human resolving power, just to discuss, we covered this in the class. So resolving power is basically whether you can differentiate between two objects. So if the two objects are clearly visible, they are well resolved. And if they are merging like this, or they appear as a single peak, they are not resolved. So the human eye, it can only resolve up to 0.25 millimeter. Beyond that, you will either need the light microscope or so on. So, and also we saw the expression of resolving power. I'll go back to that slide. So the resolving power, the formula was lambda by 2Na, where lambda is the wavelength of the light and Na is the numerical aperture. And numerical aperture you can calculate by the ratio of diameter of the lens divided by focal length of the lens, that is D by F. So smaller the wavelength, higher will be the resolving power. That is why you can see, if we go back again, you can see the resolving power of the naked eye is only up to here, whereas light microscope is using light microscope, because you are using different lens to increase the size of the image. It has higher resolving power. And electron microscopy uses electrons which have very small wavelength, so they are able to resolve even smaller particles. For normal eye, the resolving power is only up to 0.25 millimeter. Okay. The next question is asking, what is the final detector in a cryo-electron microscope? So the answer to this is charge coupled device. So there are different kinds of detector. I'll go back again to that slide. So in cryo-electron microscopy, you can have two kinds of detectors. One is an indirect detector. So the electron in cryo-electron microscopy, after it passes through the sample, it strikes the detector, and the detector will convert the electron into, so the electron will strike the detector and a photon will be released, and the photon will be converted to an image. That is an indirect detector. But the more commonly used detector in cryo-electron microscopy is called charge coupled device, or CCD. In CCD, what happens? The electron will strike the detector and form a photon. So up to here, it is similar to the indirect detector. But then the photon is converted to an electrical signal, and this electrical signal is read by the detector and turned into an image. So that is called the charge coupled device, and that is the detector most commonly used in case of cryo-electron microscope. So it's called charge coupled device, or CCD. So that is the answer here. It is C, that is charge coupled device, or CCD. Then you have the question, surface topography can be visualized by which of the following technique?
SEM.
Correct. So we saw in SEM what happens is that you have the electron source, it passes through the lens, and it will strike the surface of the sample. By changing the orientation of the scanning coil, you can choose which region of the sample this beam is striking. So basically, you can scan over the entire sample surface. You can see electron beam, it is falling in different regions of the sample surface. So it will give you a very good idea about the sample surface topography. So that is what SEM is. And in TEM, you pass the electron beam through the sample. So TEM will give you an idea about the entire structure, like not only that of the surface. The internal structure also can be determined using TEM. So since here it is asking surface topography, the answer is SEM.
So the image is about the pollen grains, is of the pollen
I
grains.
am not sure actually. It's probably pollen grains as far as I remember, but I have to check once. You can see
Okay.
only the surface can be clearly seen.
Yes, ma'am.
So yeah, it is using SEM. Then very simple question, that what is the full form of PDB?
B, option B.
Protein data bank. As you know, all the protein structures are deposited in the PDB. Then the whole virus can be best viewed using what?
Cryo-electron
Yeah.
microscope.
So viruses we have seen are very small. You can see the viruses are around 0.05 to 0.1 micrometer. That is beyond the range of the light microscope, and only an electron microscope can be used to visualize viruses. So the answer is C, that is cryo-electron microscope. Then it is asking, which of the following term is used for rapid freezing of the sample at a rate of 10 to the power 6 degrees Celsius per second. So the water in and surrounding the specimen is fixed in a vitreous state.
Is it freeze etching? or vitrification?
No, actually it is vitrification. So I'll like I'll explain. So vitreous state basically means you are freezing the water in the native orientation. So I'll just discuss this. You have the living tissue composed of water. Now, if you are slowly cooling the tissue, water will form this kind of hexagonal ice crystals. And the hexagonal ice crystal will completely destroy the structure of the cell. But if you are freezing it very fast, like at this fast of a rate, then the water does not have time to form this kind of a crystal. It will just be frozen in the native orientation. And so in the native, the cell is also maintained in the native orientation. It does not destroy the cell structure. So water in this state is called crystalline water, and water in this state is called vitreous water. And so it is called vitrification because you are freezing the water in a vitreous state. That is option D.
Ma'am, can you explain it once more?
I'll go back also to, I'll see if my previous slides are better. See, when you are freezing the sample very slowly, ice crystals form, like the hexagonal ice crystal that I was showing. And when ice crystal form, it will destroy the structure of the cell. The cell will completely collapse. So that is called crystalline ice. And in vitrification, what happens? So imagine these dots are the water molecules. These pink dots are the water molecules. In vitrification, what happens is that you are freezing the sample very fast. So the water does not have time to form this kind of hexagonal ice crystals. Wherever the water is, it will just freeze in that orientation. And so it will not destroy the structure of the cell. The cell is maintained in the native state, and the water is just frozen in the orientation that it occupied. This kind of state of the water is called a vitreous state. And if it forms this kind of hexagonal crystals, it is called crystalline state. In the crystalline state, it will destroy the cell structure, so you will not get a good image. But in the vitreous state, the water is just freezing wherever it is. So the cell structure is maintained and you will get a good image. So this kind of process where you are freezing very fast to freeze the water in a vitreous state, it is called vitrification. That is option D.
So where freeze etching technique is used?
Okay, that I have to search actually. Freeze etching is not a word that is associated with cryo-electron microscopy. So I have not looked into it. I'll look into it and let you know.
Okay.
Then it is saying the internal structure of a bacteria can be best viewed by which of the following technique?
TEM?
Yeah, that is correct. So again, the difference between SEM and TEM. In SEM, you are focusing the electron beam on the sample surface. And in TEM, you are focusing the electron beam to pass through a slice of the sample. So SEM will only give you idea about the external structure, whereas TEM will tell you about the internal structure of the bacteria also. But one disadvantage of TEM is that this sample slice has to be very thin, because if it is a thick slice, the electron beam will not pass through it. So that is one disadvantage. But if you can manage to get a very thin slice, then you will get a clear structure of the internal structure of the bacteria. So both compound microscope and bright field microscope use light. They are light microscope, so they are not very good for visualizing bacteria. SEM will only tell you about the surface structure. So TEM is the correct answer, which tells you about the internal structure of the bacteria. Okay, I'll go to the next question, which is saying, which is the first 3D electron microscopy structure released in the PDB? So this is also like an information question. It is actually bacteriorhodopsin. So this is something you just have to remember. Like the first protein structure determined was myoglobin, which was determined by X-ray crystallography. The first electron microscopy structure determined was bacteriorhodopsin. So you just remember this as a information. Then it is asking that how many years have we been celebrating PDB on 2021? Again, just an information question. It has been 50 years. So it was first started in 1971. So from there to 2021, it has been 50 years. Again, this is an information question. It is not like any explanation or anything. And as you can see, over the years, gradually the number of structures released has exponentially increased. And initially, the structures were mostly determined from X-ray crystallography, but now more and more structures are also being determined using cryo-electron microscope. The next question is asking the power of an objective lens to separately distinct two adjacent points is known as what?
Resolving power.
Yeah, we just saw that. It's resolving power, that is B. Then it is asking the formula for the resolving power of a microscope.
Option A.
Yeah, that also we saw that resolving power of a microscope is lambda by 2 Na, where Na is the numerical aperture. And I think we have one more question for today. So long wavelength, high diffraction and less resolution are characteristics of what?
Both red and blue light.
So actually, like here it is saying come in a comparative way, right? So compared to blue light, red light has longer wavelength, and we know longer the wavelength, less will be the resolution. So in that case, it is red light that is the answer. So here it is actually because it is you are right that compared to electrons, definitely both red and blue light have low resolution. But here the question is comparing between red and blue light. So you have to say the answer is red light. So that is the questions I have for today. So today I've mostly covered, I started from light microscope, then compound microscope, and then went into cryo-electron microscope. And in cryo-electron microscope, I discussed the different ways of sample preparation, the instrumentation, the detection, and finally the image reconstruction to get a 3D image. So that is what this class has been about. And as always, I'll upload the video and the slides and you can go through them. And you were asking about NMR, so you can go through the week 6 video also. And you can just ask me questions from any of the weeks. Yeah, you can ask next class or you can email me your questions. So I think I'll end the class now. A little bit early, I'll end it. Okay.
Thank you, ma'am.
Thank you. Thank you.